Alright, so chapter 17 covers the endocrine system. The endocrine system is one of my favorite systems. It's the other major controller in the body. So the nervous system is the fast controller, gets immediate responses going. But the endocrine system is the long-term controller that controls longer-term homeostatic variables such as reproduction and feeding. So it is also very important. It's a phenomenal system. So today we're just going to get through our overview. We'll pick up with anatomy on Thursday. So we'll talk about different types of glands, how cells communicate. We'll compare our nervous and endocrine systems. And we'll talk in general about hormones. So glands. We've got endocrine and exocrine glands. We talked about exocrine glands last semester. Endocrine glands are glands. Endo means within. Crine means to secrete. So endocrine glands secrete within the system. So they synthesize hormones and secrete these hormones into the blood. Endocrine glands are also commonly called ductless glands. So examples of endocrine glands are like your thyroid gland that sits in the front of your throat, or your adrenal gland that sits on top of your kidneys. Exocrine glands contain ducts and they secrete their products onto an epithelial surface. So we met those last semester when we talked about the different types of exocrine glands of the skin. Sebaceous glands, sudoriferous glands. All of those are going to be glands that secrete their product into ducts that dump onto the surface. We will talk about some exocrine glands when we get to the digestive system, but what we're talking about in this chapter are endocrine glands, these discrete organs that are going to synthesize hormones and release them into the blood. So that's an important part, that the hormones are being released into the blood. Is everybody good on that? Okay. So, what are the different ways that cells communicate? So, endocrine glands, or cells that are secreting by endocrine secretion, we'll say that there's endocrine secretion, or we can communicate by the endocrine method, and this is going to happen across long distances. We've got a cell, and he's got a message, so that says memo, and he's got to get it out. And in endocrine signaling, this message has to go across a long distance to get to the target cell, who's sitting here waiting to receive this message. So this message is the hormone that's being dropped this into the blood, and then it's going to travel a long way to get to its target cell. If hormones are being dumped into the blood, then that means that every cell in the body is exposed to a hormone that's in your blood. So what do you think makes a cell specific, or a target cell for a specific hormone? Receptors. The receptors that it has. So it's got to have the right receptor, or the right address. So this memo is only going to one place, and this message is only going to one cell, and that cell will have a specific receptor. So there's this receptor ligand specificity in endocrine signaling. So that's going to be what we're talking about most of this chapter, is endocrine signaling. But cells can communicate by other methods. So last semester we talked about neurocrine signaling. So with the exception of neuroendocrine signaling, neurocrine signaling is when you've got one cell and he's got a message and it's just going to go a very short distance to another cell. And again, this cell has to be a specific target for that message. It can only pick up that neurotransmitter and respond if it's got a receptor for that neurotransmitter. So neurocrine signaling is across a very short distance, the synapse. So your sending cell would be a neuron, and your receiving cell could be another neuron. It could be a muscle cell, or it can be a gland. So that's what we're going to talk about in this chapter, is what our neurons the neurocrine signaling that we'll talk about is when neurons are affecting our glands. So neurocrine signaling is going to be just this short distance signaling that happens across the synapse. Now, this gland here, if we're releasing a neurotransmitter from a neuron and it's entering the blood and going far away to hit the gland, then that's neuroendocrine signaling, which is like a mix. So when we're talking about a neuron hitting a gland, that would be like in your autonomic nervous system. When we talked about the sympathetic nervous system, we said that you could directly stimulate your adrenal gland to release epinephrine and norepinephrine. That's going to be short distance signaling, where the neuron is stimulating that glandular tissue to release epinephrine and norepinephrine. So those are going to be our targets of neurocrine signaling, and that's going to be across a short distance. There's another type of signaling that happens across a short distance called paracrine signaling. So paracrine signaling affects, it's across a short distance, and it affects every cell in the area that has a receptor for that chemical, for the chemical that's being released. So we'll say paracrine is across a short distance, and it affects every cell with a receptor. So what would this look like? If we had a cell, a sending cell, that just throws out a bunch of messages to the whole general area, just takes its paper and throws it. If you've got a receptor for it and can catch it, that's paracrine signaling. So we'll talk about paracrine signaling later in the semester. The last type of signaling that we have is called autocrine. Auto always means self. So autocrine signaling is a message to yourself. So it's like this guy's got a note to self. And this happens, we'll see, in like the immune system. Let's say you get infected with a virus, then you can release these autocrine signals that will come back and bind, and that will help stimulate you to start an immune response or to beef up your antiviral properties or something like that. So autocrine is going to be self-signaling, and we'll see that as we move throughout the semester as well. So endocrine is across long distances. Neurocrine is across a very short distance, the synapse. Paracrine is just like a general message to the area. And an autocrine signal is when a cell releases a chemical that is then going to bind back to the cell and create some change in the cell. Are there any questions on the different communication types? Okay, so if we look at our nervous system communication method and compare it to our endocrine system communication method, then what are the similarities and differences? So these are our two main controllers. In the nervous system, we've got neurons that are going to hit their target cells, and they're going to use neurotransmitters. In the endocrine system, we've got some gland that's releasing a hormone to the blood. And the target cell can be very far away. So the chemical here is a hormone that's being released to the blood, and it's got to travel a very long distance to get to its target. So a difference would be then the distance of travel or distance that the message has to cross. They're both using chemical signals. Neurons are using neurotransmitters. Glands are using hormones. A similarity is going to be that both of our target cells must contain receptors for the chemical. So target cell specificity is something that the two systems have in common. That's pretty much it for the comparison. What else could we say? That the nervous system is quick. So responses in the nervous system are going to happen very fast. The endocrine system is slower. These are going to be longer-term homeostatic variables. And it kind of makes sense. If you've got to go from your pituitary gland to your ovaries, there's a long distance to be traveled. So the response is going to be slower. This is quick. These are also, the response happens quickly, but these also end quickly. So when you're done stimulating this presynaptic neuron, then you will use enzymes to break down the neurotransmitter or reuptake it, and so you can stop the signaling pretty quickly. So we'll say this stops quickly. In the endocrine system, it can't stop that quickly. You can't immediately take it back. You've got a hormone floating around in the blood. So the responses are, they're much longer lasting. So we'll say they're longer lasting effects in the endocrine system. Other things, other comparisons, we could say that the targets of neurotransmitters will always be neurons, muscle fibers, or glands. So those are always going to be the targets. The targets in the endocrine system are any cell in the body with a receptor, so they can be very widespread effects. And in the case of like your stress hormone, cortisol, every cell in your body has a receptor for cortisol. Now, they may have different affinities for binding cortisol, but eventually every cell in your body can respond to the stress hormone, which makes stress a dangerous physiological phenomenon. It's necessary in the short term to get you, to give you energy and things that you need to get away from stressful situations, but chronic stress is very bad for the system overall. Any questions on the comparison between the nervous system and the endocrine system? All right. Hormones then. Hormones are the chemicals that are being released from endocrine glands into the blood, and we can classify them a couple different ways. We can classify them based on their method of release or their chemical classification. So first let's talk about method of release. So we've got two types of hormones that we can classify based on method of release: neurohormones and regular hormones. And neurohormones are going to be hormones that are released from nervous tissue. So we'll say released from neurons into the blood. We've got two types of neurohormones. All of our hypothalamic hormones that are getting everything going up in your head are neurohormones. So these are releasing and release inhibiting hormones. So we've got releasing and release inhibiting hormones that come from the hypothalamus. Also, your pituitary gland is split into two regions, the anterior pituitary and the posterior pituitary. The posterior pituitary is nervous tissue. So they're neurons that reach down from the hypothalamus and go through this space called the infundibulum and synapse there at this area called the posterior pituitary. So these are also neurohormones, and there are only two. One called antidiuretic hormone and another called oxytocin. We'll talk more about the hormones and the specifics later. This is just our overall introduction right now. Hormones are released from glands into the blood. So regular hormones are released from glands into the blood. And we've got tropic hormones. We've got 6 tropic hormones we'll talk about. These are all coming from the anterior pituitary. And then other hormones coming from your other glands, like your thyroid glands, or your adrenal gland, or your testes, or your ovaries. So just everything else that's coming from a gland and being released to the blood is categorized as a hormone. We can also classify hormones based on their chemical classification, so what it is they're made of. Our steroid hormones have as their basis this cholesterol, and it gets modified, but they're basically starting out to give us our steroid hormones. So we'll say these start with cholesterol. Our protein hormones are chains of amino acids. Or we've got what are called biogenic amines, and these are just modified amino acids.
I'm sorry, steroid hormones start with cholesterol and nucleus?
It's called a cholesterol nucleus.
Okay.
That's what it's just this weird, it's not really a nucleus. Yeah, it's just this structure that basically you start with cholesterol and you add stuff to it, different functional groups to it, and you'll wind up with your steroid hormone.
Under that hypothalamic hormones releasing, what is that next one?
Releasing and release inhibiting hormones.
Releasing and release.
So when we go through, start looking at the anatomy and physiology, we'll talk about both types of classifying based on where it's coming from and based on the chemical classification. This is going to be more significant for how the hormone behaves. So we'll look at that in a little bit more detail. Let everybody finish getting these notes. Are we good? Okay. So as far as our chemical categories go, we've got our steroids, we've got our proteins, and we've got our biogenic amines. So our steroid hormones have that cholesterol nucleus, which makes them lipophilic. So these are lipophilic. That means that they like fats. That also makes them hydrophobic, which means that they don't like water. So what does this mean? This means that they have to be transported in the blood. The blood is an aqueous solution. So they have to be transported on a transport protein in the blood. So they require a transport protein. Now, if they're lipophilic, what about cell membranes? Do you think their receptor would be on the surface of the cell membrane, or would it be inside the cell? Our cell membrane is a phospholipid bilayer, right? So steroid hormones can slip right through it. So they work through intracellular receptors. Protein hormones, on the other hand, are hydrophilic, which means that they do not require a transport protein. They can be freely transported in the blood. Now, if they're hydrophilic, do you think that they're going to, what do you think about their receptors? Would they be able to slip through the plasma membrane? No. So they have extracellular receptors, exactly. Biogenic amines are just modified amino acids. They're very small, and so most of the time they behave like proteins. We do have an exception though. Thyroid hormone is a modified amino acid, and it behaves like a steroid. So we'll say biogenic amines most of the time behave like protein hormones. And then we'll say except thyroid hormones, which behave like steroids. Any questions so far? So for transport of steroid hormones, they're released from their gland and they have to bind a transport protein. They're transported to the target cell. They bounce off that transport protein, slip through the cell membrane, and bind intracellular receptors. Protein hormones can freely mix in the blood, so they're just released and they flow down into the blood. They'll bind extracellular receptors, which means that they've got to be able to stimulate. These extracellular receptors have to be able to stimulate an intracellular response. So we'll look at that in just a minute. And then our biogenic amines, most of the time are going to behave like proteins with the exception of our thyroid hormones, which act like steroids. All right, so as far as transport and actions go, our water-soluble hormones, which are our proteins and most of our biogenic amines, readily dissolve in the plasma. They're going to bind surface receptors and activate second messengers. Second messengers are chemicals inside the cell that are going to carry out the response of that extracellular receptor. Our lipid-soluble hormones, which are our steroid hormones and thyroid hormones, do not readily dissolve in the plasma, and they bind intracellular receptors, and what they're going to do is alter gene transcription in some way. All right, so our lipid-soluble hormones, which are our steroid hormones and our thyroid hormones, are going to be released and bound to a transport protein, travel through the blood, they get to their target cell and can slide right through this phospholipid bilayer, and then they're going to bind with an intracellular receptor. So it would be specific for our hormone. So the ligand receptor specificity is important. So for our little hormones, this square guy, it can bind its intracellular receptor. And now this intracellular receptor is actually going to go into the nucleus and interact with the DNA on what are called hormone response elements. So our hormone receptor complex is going to bind our DNA and alter gene transcription. So this this is the ligand receptor complex, and it's binding to the hormone response element, or HRE. The hormone, that's not going to line up right. Just be aware, the hormone response element is going to be responsive to the particular hormone. So if it's testosterone, it's the testosterone response element. And so the receptor comes in and binds your DNA and alters gene transcription. So this could be like a muscle cell, and you would increase muscle protein production then in males if it was responding to testosterone. Increase hair production, things like that. So steroid hormones are altering gene transcription, so their effects are definitely long-term. Another example, estrogen. Estrogen binds the estrogen response element on DNA and causes increased thickening of your uterine wall. You increase mitosis in the endometrium of the uterus so that you get thickening of that wall. Progesterone binds the progesterone response element and causes production of new blood vessels in that thickened endometrium. So our lipid-soluble hormones are going to have longer-term metabolic effects than or longer-term effects than our water-soluble hormones. And that's because they're interacting with the DNA and altering gene transcription. our water-soluble hormones are going to do something different. They're going to activate what are called G proteins. So if we look at our water-soluble hormones, or our protein hormones, and most of our biogenic amines, they're going to bind to an extracellular receptor. This extracellular receptor is what's called the G protein coupled receptor. And when you see a G protein coupled receptor, they always have these 7 transmembrane spanning domains. 7. And so your book, I think, just shows them as blobs. Some books you'll see the 7 transmembrane spanning domains, and they're associated with this G protein. It's called the G protein coupled receptor. When they bind, they're going to activate another protein that's found in the cell membrane and are going to start kicking out our second messengers. Your book has the specifics of 2. One that's called the cyclic AMP pathway, and another called the IP3-DAG pathway. If you're going on to medical school, you must know the specifics of both of these pathways, so study them. But for our class, we don't have to know the specifics of them. I need you to know in general what second messengers are and how it is that they're working. So what happens when you stimulate this G protein coupled receptor, it's going to stimulate this other protein to start kicking out one of these second messengers. And then these second messengers can go off and do many things inside the cell. They can, let's say, turn on or turn off enzymes. They can also alter gene transcription. So your second messenger may be able to go on and alter gene transcription. But ultimately what you're doing then is the first messenger is the hormone binding its extracellular receptor. It activates this G protein coupled receptor, and you start kicking out these second messengers, and the second messengers are going to be able to carry out the message inside the cell. They amplify the message inside the cell. So as long as this receptor is bound out here, you can be making second messengers in here, and then they can go about and do a whole bunch of different stuff inside the cell. So the IP3-DAG pathway is going to be one that gets a whole bunch of intracellular calcium. So in that case, calcium would be the third messenger. So if you remember from last semester, calcium is a huge signaling molecule. So we can actually use this IP3-DAG pathway to open up calcium inside the cell, and then calcium acts as a third messenger. Again, I'm not going to worry about the specifics of this, but I would want you to know in general that our protein and most of our biogenic amine hormones are using G protein coupled receptors that are activating second messengers. So I could ask you something like, second messengers can do what in the cell? A, turn on an enzyme. B, turn off an enzyme. C, alter gene transcription. D, all of the above. A or E, A and B only. And you'd say all of the above. So just know in general how second messengers are working. Also, if I were to ask you, well, actually I think I have a clicker question, so I won't ask you. I'll give you the clicker question. So again, you've got the 2 different pathways shown in your book. You can look them over. I'm not going to test you on the specifics, but if you go for the MCAT or something, definitely know the specifics then. Second messengers can do many, many things inside the cell. So they can turn on or off an enzyme. If you've got a stored hormone sitting in a storage vesicle, like our thyroid hormones, then when our thyroid stimulating hormone binds, the second messenger can cause these vesicles to release thyroid hormone right to the blood. So second messengers are going to be pretty quick, because when we bind that extracellular receptor and get those second messengers going, then we can have immediate responses. You can also see longer term responses by altering gene transcription and things, but you can also see immediate.